Ladies and gentlemen, welcome to the seminar on Life Science and Medicine of Masterminds, Masterclasses 2.0, organized by the Council of Hong Kong Laureate Forum, also known as the HKLF. First of all, allow me to introduce myself. My name is Tasha Zheng, and it's my great pleasure to be your MC today. And on behalf of HKLF, a very warm welcome to each and every one of you joining us in person today. And we'd like to extend our heartiest welcome to our online attendees joining us from all around the world on the live webinar platform. Thank you for joining us today. In the last 2 days, we have had enlightening sections and discussions with different experts and scientists on different topics. And today, we're very honored to have invited 3 renowned scientists with research interests in life science and medicine to share with us their captivating research. They are Professor Gero Miesenbach, who will be joining us from the US. And we also have Professor Alice Wong from the University of Hong Kong and Professor Zhang Jianhua from Hong Kong Baptist University joining us in person today. There will be a Q&A section after each of their presentations for you to ask questions and exchange ideas with them. We invite you to raise questions from both our live audience and our online audience. For our online friends joining us virtually, feel free to use the Q&A function on the live webinar platform. And to facilitate today's seminar, in particular the Q&A sections, we are very delighted to have Dr. Stephanie Ma, Associate Professor of School of Biomedical Sciences at the University of Hong Kong, to be our moderator. Without further ado, may we now invite Dr. Ma to give an opening speech and to introduce the first speaker for us. Dr. Ma, please.
Thank you, Tasha, for the very nice introduction. Good morning, ladies and gentlemen. My name is Stephanie from the University of Hong Kong. It's my pleasure to be here this morning to be the moderator of this session on seminars in life science and medicine for the Masterminds Masterclasses 2.0. So it's very nice to see so many faces and so many young faces in the audience, and it's really a very nice chance because we have the pleasure, and that's our seminar today is graced by the presence of 3 very distinguished speakers. But before we proceed to the speakers, I have been asked by the organizer to introduce a little bit about myself first and to tell you a little bit about my research journey and how I started in science. So as Tasha mentioned, I am working at the School of Biomedical Sciences, Faculty of Medicine at the University of Hong Kong. So there I actually teach an undergraduate program in the biomedical sciences, and I'm also involved in teaching of the Year 1 and Year 2 MBBS program, where they start learning about the basic biology, anatomy before they go into clinical rotations. But more importantly, and more interestingly for me, I actually also run a cancer research lab at Hong Kong U. And as you know, cancer is a very deadly disease. It's also a very tricky disease. The tumor itself, it's made up of many different cell types. And we are particularly interested in a particular type of cells in the tumor that is called cancer stem cells. So cancer stem cells, if you hear stem cells, you probably ring a bell that you hear in the news. It's for anti-aging or for rejuvenation or something good to make you, to beautify yourself or something. But cancer stem cells is actually the bad guys. It's the bad guys in the cancer, and we have now shown that it actually is responsible for tumor recurrence and also for therapy resistance in tumor. And so over time, even if the cancer can be shrunk by standard therapy, because of the unique abilities of these stem cells to self-renew and differentiate, they are able to repopulate the entire tumor. So it's basically, we believe it's the root of the disease. So in the lab, we're very passionate about trying to understand more about cancer stem cells, how we can better target them, and it is really our wish to hope to find better therapy to target cancer stem cells for precision medicine, and also more importantly, to try to find new biomarkers for earlier diagnosis or even prognosis of the disease. So cancer, of course, is, as I mentioned, is something that I think a lot of us hold close to our heart. We have lost loved ones from cancer. We know directly or indirectly someone that has been affected by cancer. And unfortunately, it is in the clinic, there is still a large clinical unmet need. So we really, in the lab, is very passionate to find something for a solution to cancer, hopefully in the future. And I think for young ones that are to start off and are interested in looking for science, it is very, very important that you actually identify a topic that you also find very important, that you also find that is close to your heart. And also that it's very important to find to know the knowledge gap, that there is a knowledge gap in the topic, and that you someday would also find a solution for. So if you have that passion, and if you find this passion, trust me, you will never work in a day in your life, because going to work is just like trying to solve a mystery, for me at least, right? So it's like, you're learning every day, you're exploring every day, and it's on a topic that you find interesting. So you won't find it tiring or going to work. So enough about me, and so I would want to switch gears and to really focus on our distinguished speakers. So I would like to invite our first speaker, who is Professor Gero Mesenberg. So Professor Mesenberg is actually currently the Wainfleet Professor of Physiology and Wellcome Investigator of Center for Neural Circuits and Behavior at the University of Oxford. So Professor Mesenberg actually started studying medicine at University of Innsbruck in his native Austria, and then went on to do his postdoctoral training in New York at the Memorial Sloan Kettering Cancer Center. He actually made faculty at Memorial Sloan Kettering Cancer Center and also at Yale University before before going to Oxford to take up a faculty position in 2007, where he is still currently at. So Professor Mesenberg is well-renowned and also recognized by many, many awards and honors over the years. And just to name a few, the more recent ones includes the 2019 Rumford Prize, the 2020 Shaw Prize in Life Sciences, as well as the more recent 2022 Louisa Gross Horwitz Prize. Dr. Mesenberg is a pioneer in the field of optogenetics, which features basically the ability to use light to turn on and also to turn off brain cells. And today he will be sharing a talk that is entitled A Game of Chance. So with that, I will pass the stage to Professor Mesenberg. Are you here? Hello.
Yes, hello, Stephanie. Can everybody hear me?
Yes, so the stage is all yours, Professor Mesenberg.
Thank you very much for the kind introduction. I'm actually joining you from Los Angeles on the west coast of the United States, where I'm currently on sabbatical. You're probably wondering about the title that I gave my presentation, A Game of Chance. What I refer to in this title is what we do, the process of discovery. The fact that science is a game of chance is probably the most widely misunderstood aspect of it. It's widely misunderstood by the general public, by governments, by funding agencies, by philanthropists, and even by many scientists. The point that I want to make, and I think this is the most important point of my talk today, is that the importance of our research is fundamentally unpredictable. One can either paper over this unpredictability with grand strategic visions or roadmaps, or one can hedge one's bets by making many independent investments in individuals. And this is actually how one can win at a game of chance. The argument that spreading one's bets widely is the best science strategy was made with great force more than 80 years ago by Abraham Flexner, the founding director of the Institute for Advanced Studies in Princeton. In an essay he published in Harper's Magazine under the provocative title, The Usefulness of Useless Knowledge. Flexner used many examples from the history of science and argued that because we do not know what will have practical value in the future, the pursuit of knowledge must be completely unobstructed. We can only rely on individual curiosity as our guide. The essay happened to appear in October 1939, just after the outbreak of World War II, when the pursuit of useless knowledge may have seemed disorganized and wasteful, or even frivolous. Flexner dispelled all these notions. Now, after what I've just said, I won't offer you a list of problems that neuroscientists should be working on, and instead focus on just one that piques my own curiosity. As it happened, in the very same month that Flexner published his essay, a classic paper appeared in the pages of Nature by Alan Hodgkin and Andrew Huxley, where they reported the first intracellular recording of an action potential. And you see the historic figure from that paper here, which revealed that millisecond impulses, electrical impulses, are the basic information carriers in the brain. You see these little time markers at the bottom, which are spaced in 2 millisecond intervals. So you see this is a really, really, really short event. Now, the brain uses at least 10 orders of magnitude of timescales for information processing. And action potentials, which we understand very well, occupy the very short timescale of about a millisecond. Synaptic potentials are a little longer. They last from 10 to maybe 100 milliseconds. And then there's also many slow processes that can span many years, such as the development of a human brain, circadian rhythms, which are based on changes in gene expression that occur at timescales of days, and plastic changes implicated in learning and memory, which happen at timescales of many minutes to hours. But what you see on this graph here is that there's a window spanning times from about 100 milliseconds to many minutes or hours that is relatively unexplored and poorly understood. And I would argue that at these time scales, the most interesting neurobiology happens. At these time scales happens what we are currently doing. At these time scales, we communicate. In order to understand what I just said, you have to retain a working memory of what I said a few seconds ago. And for me, in order to communicate effectively with you, I have to have a sense of the future, of the sentence that I'm speaking now, how this will evolve in the next few seconds. So this is an example of temporal information processing at the second scale. Temporal information processing at these scales is, of course, also implicated in the generation of movement sequences, such as dancing or various sports. It's important for us to be able to navigate our environments, to remember where we were a few seconds ago, and to keep a mental image of the goals that we are pursuing. And it's, I think, a general problem that underlies all of cognition. It's no accident that we refer to thinking as a train-like sequential event. The metaphor of the train of thought, which sometimes derails, as in this artwork, is no accident. It reflects the intrinsic sequential nature of thinking. Now, we study one particular aspect of temporal information processing in the brain, and one could actually say that understanding how the brain operates at these timescales that are much longer than individual action potentials will in all likelihood hold the key to many diseases of cognition, to many mental illnesses. But of course, having read Flexner's essay, which I would encourage you to do as well, and you can easily find it on the internet, I won't go there. Instead, I will stick to the basic problem that we are interested in, and that is a form of temporal information processing referred to as neuronal integration. And by integration, I don't mean some woolly form of this brain area integrates signals with that come from other brain structures, but integration in its mathematical sense, as the accumulation of a mathematical function over time, the computation of long-term averages of signals that accrue over time. And we address this problem in two specific behavioral contexts that span, once again, different timescales. The first is the problem of perceptual decision-making, where we know that decision makers improve their choices by accumulating sensory evidence over extended time frames. And it's known that the more difficult a decision, the longer it usually takes us to make them in order to gather enough information. Now, this process typically takes on the order of a few hundred milliseconds to several seconds. So again, much longer than the impulse of an individual action potential. So the question then is, how does the brain actually accumulate evidence over time? How does it retain the intermediates of computation, and how does it discharge behavior once the response threshold is met? So this is one context for our research. The other one is the regulation of sleep and waking. And you can see that this problem is conceptually quite similar to that of decision-making, because once again, there is a temporal integration process that underpins it. Instead of sensory evidence, what accumulates in sleep homeostasis is something called sleep pressure. And when that something, and I'll give you a hint of what that something is in a second, reaches a certain threshold, we go to sleep. So I'll give you just a brief glimpse of the kinds of answers that we have obtained as to how the biophysics of the brain and of the nervous system actually solves these mathematical problems. The answer to the problem of sensory evidence accumulation in perceptual decision-making, at least in the system that we study in fruit flies, is that they have a specialized class of neurons which act as accumulators because they have a very long memory for voltage changes. And these neurons, as you see on the right, actually implement this process of temporal integration of sensory evidence by collecting synaptic quanta in a stepwise fashion. And these quanta gradually push the membrane potential of these neurons, which at resting level is very far from spike threshold, up to spike threshold. And once that spike threshold is reached, and these neurons discharge an action potential, just like the spike that Hodgkin and Huxley recorded, the decision is complete and the animal commits to its choice. So this is a process that, as I mentioned before, happens at relatively short time scales of hundreds of milliseconds to seconds, whereas homeostatic sleep control is much slower. It happens during cycles of sleep and waking, where the sleep homeostat, referred to also in engineering terms as a relaxation oscillator, fills with something. This is the accumulation process. And when that something has reached a certain threshold, the system tips from fill into discharge mode. The accumulated sleep pressure is released during sleep, and the animal wakes up, and then the process begins anew. So this usually takes, depending on the animal species, minutes to hours to complete a cycle. But again, in order to be translated into behavior, it ultimately leads to a change in the electrical activity of neurons. So a change in these very fast millisecond events. But the discharge of these fast events has to be regulated by much slower processes. Now, these processes we've discovered have to do with how efficiently neurons in the brain burn calories from food. Sleep-inducing neurons have a particular sensor, which you see at the top of these slides, a potassium channel called shaker, which has a so-called beta subunit, Kv beta, which hangs under it like the gondola under a hot air balloon. And that Kv beta subunit has a small molecule bound, which is in its reduced form, NADPH, when the mitochondria, which you see below, burn calories efficiently. So when every electron that enters the mitochondrial transport chain is actually used for ATP synthesis, and supply and demand are in perfect equipoise, the sensor at the plasma membrane keeps the NADPH in its reduced form. But if there's an excess of calories flowing into the mitochondrial electron transport chain, as you can see here, the transport chain creates something called reactive oxygen species, such as the superoxide molecules that you see coming out of complex 3 in this case. And these molecules indirectly trigger a change in the cofactor bound to the Kv beta subunit and lead to its oxidation from NADPH to NADP+. And what that does is it actually changes the electrical activity of the sleep control neurons. So this is a mechanism that allows a slow process, such as, or a biochemical process, such as mitochondrial respiration, to be transduced into changes in electrical activity. You can see in these recordings from the same neurons that when NADPH is bound to the sensor, these neurons produce relatively few of these electrical events, whereas if NADP+ is bound, the spike frequency increases quite dramatically. And it's this increase in spike frequency that is the mechanism that transduces sleep pressure into sleep. And when the animal is sleeping, the bound NADP+ is actually released by the electrical activity of these sleep-inducing neurons and replaced again by NADPH. So what I've described to you then is a biochemical memory cell that consists of two components: an ion channel that is voltage gated and consists of the Kv alpha subunit, and a redox sensor called Kv beta. And these two components are coupled in a firm association in this ion channel at the plasma membrane. And this tight coupling of two components is reminiscent of a semiconductor memory cell, like the ones that are present in our cell phones and in our computers. This is the classic dynamic RAM that's used as working memory in most of our electronic devices. These dynamic random access memory cells also have two components. They have a storage capacitor where 1 bit of information is held in the form of a charge, and an access transistor whose voltage regulates whether information can be written or read from the storage capacitor. So we think that this voltage-gated potassium channel, which, with its two components, plays exactly the same function, that the beta subunit is the storage capacitor which holds 1 bit of information encoded in whether it's NADPH or NADP+ that's bound. There's your 1 bit. And the voltage across the membrane spanning protein and its voltage sensors actually determines whether NADP+ can be exchanged for NADPH, so whether information can be written or released. So there's in fact two inputs to this device, just like to a random access memory in a semiconductor. One of these inputs is the efficiency of mitochondrial respiration, whereas the other is the electrical activity of the sleep-inducing neurons. So we have reached a stage where we can replace the conceptual animation that I've showed you before, where I've described the relaxation oscillation that consists of a vessel filling, then tipping, and then emptying, and this process repeating over and over again, with a molecular picture of what is actually happening in these sleep-inducing neurons. And here is that molecular picture. You see in the membrane of this cell yellow channels that I've not talked about. And these yellow channels can either be at the plasma membrane, or, as has happened just now, they can be internalized in intracellular storage vesicles. Now, these yellow channels, which are called Sandman, act like a brake on the electrical activity, and they correspond to the situation where the vessel that we saw before is either tilted or upright. When the channels are in the cell interior, the vessel is in discharge mode, whereas when they're on the plasma membrane, as is happening right now, the vessel fills. Now, what does it fill with? You can see that the Kv beta subunit of the voltage-gated potassium channel progressively fills with NADP+. Now the process is starting. So you see that during waking, NADPH is gradually replaced by NADP+ as the mitochondria generate reactive oxygen species. Once sleep is induced and the neurons become electrically active, the bound NADP+ can be exchanged for the more abundant NADPH, and the system resets itself during sleep. This brings me to the conclusion of my presentation today. I hope I've given you at least a flavor of the kind of problems and the types of biophysical answers that we hope to obtain to them. But if there's one thing that I would ask you to bear in mind after today, it is Abraham Flexner and his plea that it's often the seemingly useless knowledge that turns out to be the most useful. Thank you.
Thank you. Thank you very much, Professor Miesenberg, for your very informative presentation on temporal information processing. May I now invite Dr. Ma to come on stage again for a Q&A section. Dr. Ma, please.
Thank you, Tasha. Thank you, Professor Miesenberg, for the very intriguing talk. We have a few minutes for Q&A, and I have seen some questions that have popped up online. So maybe I'll start by asking one of them. So as Professor Miesenberg mentioned, optogenetics is, of course, a very powerful technique that can control cell states and cell functions or their cell actions. So may I ask whether this technique is it being used, at least, or have it been used in the treatment of disease at the moment?
The short answer is yes. Optogenetics is being used in clinical trials, in studies trying to restore vision. There's obviously the potential to intervene in nervous system function in many different contexts, but I think that the retina is the right place to start. Because there are significant technical, ethical, and also intellectual hurdles to optogenetic applications in, for example, cognitive processes. The retina is immunologically privileged. It's relatively easily accessible. And most importantly, we know exactly what it does. There are diseases where photoreceptors die, and the ability perhaps to reintroduce light-sensitive elements downstream in the circuitry of the retina can restore at least a modicum of vision.
Thank you. So it's good to know that things that we actually do in the basic research lab is being applied and being used. I have another question from the audience mentioning that, because you showed actually a math modeling in your presentation, and I guess the question is, if we're in the life sciences field or in the biological sciences field, it's knowledge in mathematics and also in computational mathematics a very, very important element in life sciences. Yes,
absolutely. Absolutely. So if I always joke that I wasted my youth by studying medicine. If I could live my life again, I would probably still end up being a neuroscientist, but I would probably begin with an undergraduate training in physics and mathematics.
That's very interesting.
I think these disciplines give you a principled outlook on the way the world works. I think one of the most important things to remember today, where big data are becoming increasingly prominent in biology, is that understanding rarely falls out of data collection. Understanding usually requires that you have a model of how you think the system works, that you make an educated guess, just like we did in all these studies that I talked about, that you articulate a clear hypothesis, which may be wrong, but that can guide your experimental work.
Just to follow up on that, you know, I think it's very intriguing how you studied medicine, but then you went on a very successful scientific career. But what was that initial switch that made you to abandon medicine, or clinical medicine at least, and to become what are you doing now? Because I think in people in Hong Kong, medicine is a very obvious choice if they have the grades, and not a lot of people do pursue scientific career if they have the grades to get into medicine. But I really want to hear from your story how to inspire the younger generations now.
I mean, I don't want to be flippant, but I mean, one advantage of science is that you can repeat a failed experiment, whereas in medicine, that is not always possible. I think that there was also sort of an intellectual difference. In science, you sort of seek the uncertainty, you seek the unknown, and that is something that suited me temperamentally. Whereas in medicine, if you want to become a really, really good clinician, you have to become almost automatic in many of the things that you do, and many of the decision processes that you run through. You sort of have to embody the knowledge that you've absorbed during your studies. You mentioned that in Hong Kong, many people pursue medical careers when they have the grades, and I sort of encounter similar things in Oxford. So when I speak to students, many of them say, actually, my great passion is music, but I had the grades, so I felt I had to study medicine. And I think, and you said that yourself, Stephanie, in your introduction, that the passion for what one does is something extremely precious and valuable, and something worth pursuing.
Thank you very much, Professor Muizenberg. So that's very enlightening, and I thank you for joining us overseas, and we'll move on to the next speaker, and thank you very much, Muizenberg.
The pleasure and the privilege. Thank you.
So it's my pleasure to introduce our next distinguished speaker, who is Professor Alice Wong from the University of Hong Kong. Professor Wong is currently the Associate Vice President of Research at the University of Hong Kong. She's also the Director and also the Professor in the School of Biological Sciences in the Faculty of Medicine at the University of Hong Kong. Professor Wong's research interest is on signal transduction in cancer, and primarily in using ovarian cancer as a model system. She's very renowned in her area, and she has been graced by many awards, including the Croucher Senior Research Fellowship, and more recently, the RGC Senior Research Fellow Scheme. So today, Professor Wong will be sharing her talk entitled Cancer Cells in Motion, Capturing Adhesion Dynamics. So without further ado, Professor Alice Wong, please.
Thank you, Stephanie, for the very nice introduction, and thank you for the Hong Kong Laureate Forum for inviting me. It's a great honor to speak here, and as Stephanie mentioned, it's very nice to see many young students here in Hong Kong like science and want to come to learn more about science. So I do cancer research, and I'll talk something about our more about our recent research on cancer cells emotion. So we have been switching from understanding cancer from a static view, that means non-moving view, to when it moves. So which we find something interesting. And I'm in the School of Biological Sciences of Faculty of Science, and this is actually the building of my lab. So if you come to Hong Kong, you do come and visit my lab, which my lab probably is hidden behind the trees like in this picture. So cancer, as we all know, is one of the leading cause of death, and in some countries, actually, it is the top one cause of death. And of all, like most, if not all, cancer types, metastases is actually the reason why causing deaths in patients. And it is about like 90% of the cancer cases, the patients are died because of metastases. Why it is so lethal? There are different reasons. First of all, it's diagnosed late. I'll talk a little bit more later on. But also, there is a lack of drugs and drug targets. So the current drugs, most of them, as you know, they are targeting the proliferating cells. So might not be very specific to the metastatic process. So there is a need for understanding the metastatic process as well as in a way so that we can have new targets and new treatment. As Stephanie mentioned, I've been using ovarian cancer as a model a lot. So ovarian cancer is one of the most common type of women's cancer, even not the most common one, but it is the most lethal one. It causes the highest mortality among all the gynecological cancers. So it has several issues. It doesn't have a good early diagnostic test. And it is in fact known as the silent killer. So more than 70% of the cases, when they are found, they are already at very late stages. So when they are at late stages, as you can see, that basically the prognosis or the treatment options are very few. You cannot do a surgery at this point. So quite lethal, and they are also resistant to a lot of the drugs. This is a cancer statistics from the US. I like this figure as it shows something to us. Because if you look at it, it's spanning across about 80 years in the history. So if you, they show many cancers, but if you look at some of the women's cancers, like the breast, or even like the uterine, or maybe even the colorectal, a lot of them, there is a decline already in terms of the death rates throughout the years. So with scientific research, it has been helped a lot. But if you look at ovarian cancer, basically in the past like 50 years or 80 years or even 100 years, very low changes in the death rates. And in terms of incidence, and in China, it is also increasing. The mortality trend in China is similar. They doesn't have, China doesn't has for a long like analysis, but even in the recent 30 years, you can see that it is like, no change at all. So why there is no changes? There are several reasons for that. So I'm just showing you a picture of mine. So I started to learn about ovarian cancer and about cell adhesion molecules, which I'm going to talk about today, when I started my PhD at UBC with my supervisor, Professor Nellie Alsberg. She's actually the one that do a lot of work on the precursor cells, and also Dr. Peter Liang. So I want to talk about cell adhesion molecules. This is actually one of the staining of the cancer cells that I did when I was a PhD. So you can see that between the cells, when they come in contact, they are like the glue. So you just like spreading the glue so that they stick together. And you can see very nice staining between these cell junctions. And why using ovarian cancer as a model? I already mentioned a little bit. But in terms of metastases, which is my focus after I started my own lab, because the patients, as I said, a lot of the time asymptomatic, and they build up these ascites, these acidic fluid, which inside you will see these single or cells in clusters. Indeed, a lot of the time these clusters or cells in spheroidal or this shape, they survive much better than some of the single cells. They take up a very unique metastatic pathway. So unlike blood metastases that usually go via the blood, they spread within the peritoneum, which they disseminate from the ovary as single cells or in clusters, and then finally settle on the peritoneum, the wall, mainly the mesothelium cells, which I'll talk more about that. So when I first started the lab, I have looked into the adhesion molecules, trying to see the changes at different point of time, and more using a static way at that time. And I'll tell you more about what we find later on. So before I go into the new animal models that I built, I want to tell you that why cancer or metastasis is so difficult to understand. Even up till now, we haven't had a very clear cure treatment yet, because it is heterogeneous. Heterogeneous come in different ways. Interpatient heterogeneity, so that's genotype, right? So each patient, they because of the difference in the genotype, even though they have the same type of cancer, they are different. And then within tumor, intra-tumor heterogeneity. So that not only comes in terms of the different cell types in the tumor. So the tumor doesn't come alone. So if you get a chance to see some of the primary tumors and get a section of it, you see different types of cells, fibroblasts, immune cells, and also some of the cell types as well. But even within the tumor cells, they are not homogeneous as well. So the drawback of the current models is, with ovarian cancer, first of all, I have to mention about the interspecies difference, because the lagging behind of ovarian cancer research is we don't have a good animal model, in particular a mouse or a rodent model, which is commonly used in the lab. The reason why is because the mouse ovarian cancer is different from the human one in terms of their phenotype and lots of the other properties. So we don't have a good animal model. And then the other, so most of the time we use human tissues and human cell lines. But as I've mentioned, with the human tissues, they have different genetic backgrounds and there are multiple cell types. If we use cancer cell lines, which has been used a lot in the lab, but they are also polyclonal. They have heterogeneity there, and there is a lack of corresponding non-metastatic control that one can use it to compare. So cancer stem cells, which has been mentioned, that is one recent discovery that helps us to understand more. So we know that in a bulk tumor, not all the cells are the same. And previously, most of the work has been focusing on the bulk tumor. So recently, we get methods to isolate cancer stem cell, which is a minute subpopulation. It can be as few as single digits, like percentage, to I think around 10. Some tumors, they might have more, like 20 or 30%, but still not the majority of it. It's a subpopulation that are these cancer stem cells. So these cancer stem cells possess the property that they can self-renew, so keep proliferating. They can differentiate to cancer stem cell or non-cancer stem cell phenotype. And then these are the ones that usually are drug resistant. And that's why leading to the tumor relapse. And we know that within these cancer stem cell populations, there is even a subset we call the metastatic cancer stem cells. So they are, again, not homogeneous. So these metastatic cancer stem cells, or metastatic initiating cells, they are the one that would land on the target site successfully and then finally form metastases. So in fact, the work, so even though a lot of those cells that actually disseminate and leave the primary tumor, you'll be surprised that less than 0.01 1% can actually form metastases. Most of them, they will just die or being eliminated throughout the process. So how can we so that's why we try to develop platforms and understanding of these very few cells that actually being successful in forming metastases. So how do we select these metastatic cancer stem cell? So we start isolating the cancer stem cell, and then we do an in vivo selection. That means in mouse, repeatedly grow the tumor first in the ovary, allow it to disseminate, and then we harvest those disseminated and the primary one, and then going through several times of the selection. So then we get quite a pure populations that are what we call the metastatic cancer stem cells, and then the non-metastatic cancer stem cells. So these are just some data showing you with the luminescence imaging. The metastatic one, they disseminate throughout the peritoneum, and then the non-metastatic one only localized in the ovary. And these are the intraperitoneal injections. So from the just a very basic RNA sequencing, you see different colors, which indicate they are quite different in terms of the gene signatures. And then these are from the patient cells. We also isolate the metastatic cancer stem cells and find that they could be very aggressive in forming metastases. And what we have been using, the cell line model, overlaps with that's what is in patient in terms of the gene signature. So showing that it's a good model that we can use to further study. But it's not sufficient, because then we realize when cells metastasize, they don't just in a static condition. They are fluid. Moving. So in the blood metastases, they go via the blood. In the peritoneum, with your gut and everything, in fact, inside there are a little bit of fluid, and they keep doing a little bit of moving. So it's just like catching a ball. So if, as I say, it's adhesion, right? So if you try to catch a ball when it is static, as compared to when you try to catch a ball when it is in motion, you will use different ways to try to precisely catch it. So at that time, when we started the experiment, nothing is known about peritoneal metastases. But there is a phenomenon in normal physiology. You all know about inflammation. So leukocyte extravasation, that means leukocyte getting to the target tissue site, actually invading it during the inflammation, is an essential and key step for this initiation of inflammation. So similarly, the adhesion to the target mesothelium is also an essential and key step for metastases to form. And in this adhesion, what you can see here is they don't just adhere. So what they do is they would have 3 steps: tethering, that means initial touching, rolling, and then adhere. And then they would repeat it several times until firm adhesion. So when there is firm adhesion, then they can invade. So we try to understand this process. And we then compare. So metastases are basically 3 major types or 2. One is through the vasculature and the lymph, and then the other is in the abdomen. So if you can look at, first of all, the target cell type is different. One is the endothelium, the other is a mesothelium. And in terms of the force, they are very different, more than 100 magnitude difference. So when with high shear stress and low shear stress, again, using very different phenomenon. So to study these in the lab, what do? We build a platform so that can capture or mimic the shear stress. So at that time, the method, or even nowadays, a lot of the methods that has been being used, like the bioreactors, the parallel plane chamber, rock place, those they cannot generate very low shear stress to study the peritoneum shear stress. So not quite physiological. And they also has uneven flow distribution, and also very difficult to precisely control the shear stress. So with a collaboration with Professor Shum, we fabricate the microfluidic chip. So it's about a device about this big with multiple channels, and with a pump to do the perfusion, and with a microscope to do real-time imaging, we could capture like the metastatic cell underflow. So we come into which I think, I'm not sure if I can do the play, or there might, so yeah, you see the motion of the green ball, which are spheroid cancer cells. So if it's playing again, you can see it. It move. Can this be played again? So move slowly here at a point, and then move again here. So they don't continuously flow. So they do the tethering, rolling, and the final firm adhesion that as I talk. So when we do the analysis real time, as I've mentioned, it's real time, we can see in brief that the metastatic stem cell compared to the non-metastatic one, they tether more, roll slower, and then finally adhere more. So showing that they do have a selective advantage for these cells. And then using the transgenic mice, we are able to map the adhesion receptor on the target mesothelium, as well as the ligand, which they are actually sugars, and then with the different branches, very unique branches that we see. And these changes in the glycogenes, we are not only observing the differences in the metastatic cancer stem cell versus the non-metastatic one, but we also see them in patients. So to continue understand these biophysical properties, as we know that, these mechanical properties are very different for the cells. So in collaboration with Professor Stella Pem, she's an expert in doing topology. That means generating gratings of different patterns. So in brief, you can see that when the non-metastatic and the metastatic cells under flat surfaces, they are very similar. But when on different gratings, the metastatic one, in terms of the speed, they already move much faster. And then in terms of the direction, it's also very unique. The non-metastatic one, they are more confined to only travel along the grooves, but the non-metastatic ones, they are more flexible. They are in their trajectory, so they are not limited by just going along the groove. So in terms of the directionality, as you can see that they are less being confined. So this has a lot to do with their actin or the adhesion molecules, which we see that the distribution is different. The non-metastatic one, quite even cytoplasmic all over. The metastatic ones, they seems to be on one edges or on one side, having more than the others. And with Professor Alfonso Ein, we look into the cell skeleton and measure the stiffness of these cells. And as you can see that again, they have different cell skeleton. So non-metastatic, they are again more even, but the metastatic one, they would have a higher cell skeleton arrangement on top of the nucleus, less on top of the nucleus and more on some of the edges. And in terms of the stiffness, they are less stiff. So that means they are softer and more flexible in doing all the movement. So talking about the tumor microenvironment, so these cells, they don't do or form metastases or be able to initiate metastases alone. They have to interact with the microenvironment. So the microenvironment has a number of different cells. And recent studies, a lot has been focused on immune cells because we know that they are being very important to help the tumor growth or facilitate the tumor progression. And we have, in particular, looked into a macrophage before I finish the talk here. So for macrophages, they are present in high abundance in the ascites. More than almost 40% of the cells are macrophages. And these macrophages are very important because experiments show that if we deplete macrophages but not other immune cells, we could inhibit peritoneal metastases. And we try, the macrophages are, again, heterogeneous. They have the M1 and the M2 type. So M1 suppresses tumor. M2 promotes it or facilitates it. So when our highly metastatic and the non-metastatic cell, when we culture them together with the M1, that means those that supposedly to suppress tumor formation, the non-metastatic one, they just die or undergo what we call senescence, or that means aging. For the highly metastatic one, they don't die. And then when we try to track them with live cell imaging, which can you help to play the video again, you will see two different arrows. The white one, as you see now, the cell divides. So this is a cancer cell. So they will undergo normal cell division. The green one is the macrophage. So you will see one coming along with the macrophage in yellow now. If you focus and look at it, it will try to divide, but doesn't separate the two cells. So by doing that, they will form a giant cell with a number of nuclei. We call the polyploidy cells. And these polyploid are important because these polyploidy cells are the one resistant to the current chemotherapeutic drugs. And they are also the aggressive one. And this can only be captured under the live imaging, because even though I said the highly metastatic cell, they don't die, but only around 20% of them, they will form these polyploid. So if we do a bulk measurement again, we will miss those very minute subpopulation. And in the patients, we again see similar polyploidy cells. So indicating what we are seeing could be very clinically relevant. So I would like to end by saying, even though we're looking at the adhesion, the glue between cells, but cells, they do communicate. So we are now trying to dissect the cell signaling mechanisms downstream of these adhesion molecules to try to better understand its mechanisms. And last but not least, I have to thank my team members. So this is one we just had last week in our lab meeting. So in particular, wants to thank Carmen, Shanshan, Aeon, Sally, Ify Samin, Katie and Jetty, who has been involved in some of the data that I showed today, and the collaborators who I have, some of them has also mentioned, and also the clinical doctors, Professor Yip and Professor Karen Chen in particular, and the funding agency. Thank you.
Thank you, Professor Wong, for the very nice sharing of your research work. It's actually very nice to be able to share the stage with you. Actually, Professor Wong and I are actually friends for a long time.
Yes, yeah.
And since I was doing my PhD, I've always been looking up to Professor Wong for her scientific accomplishments. So it's very nice to be able to share the stage with you, because this is our first time, I think.
Yeah.
So perhaps I can start by asking the very first question. So you mentioned, I think, your ability to separate the highly metastatic cancer stem cells is very intriguing. So in recent decade, right, there's a lot of sequencing breakthroughs, omics. So my question is, have you tried to understand whether that metastatic cancer stem cells that they've isolated, are they also very heterogeneous in themselves?
We tried to do some, we actually did a single cell sequencing recently on the cell line. Some heterogeneity, but quite homogeneous in a way. So I guess it also depends on how long you have been culturing them after the isolation. So first of all, is how many rounds of isolation you have gone through. Are they quite pure at that moment? And then afterwards, if you keeping them in culture, of course, like they will change under the culture condition. So if you try to keep that in a stable condition or in early passages, they are still quite homogeneous. Yeah.
So the follow-up question would be, my understanding is that these are from cell lines, right?
Yes.
So have you actually tried, or is it possible to isolate metastatic cancer stem cells from patient samples?
We did as well. Not easy, but we have a few examples, cases of those as well. So as you know that a lot of the time we get them from the SIT, so we basically gone through some of the similar procedures, yeah.
Okay. So are there any questions from the floor? Maybe I should invite.
Please raise your hand if you have a question, and we'll pass a microphone to you. Okay, if not, we can have some questions from our online audience.
Actually, I also have another question, and also from an online audience, is that we note that you also mentioned about the in vitro culture setup, right? And I understand that you have started a spin-off company based on this. So my question is, can this culture setup actually be used for cancer types other than ovarian cancer?
Yes, sure, they can. So basically, all peritoneal metastases will be similar. So if you think of what type of cancer will metastasize in the peritoneum, basically pancreatic, gastric cancers, and some colorectal cancers, all these are very aggressive as compared to some of the tumor types doing blood-borne metastases. And with the platform, as I said, the shear stress can change. Right. So if we change the shear stress, we again change the molecule that underneath that binds to it, it can be actually work for all types of tumors.
Oh, that's very useful then. Yeah. Professor Wong was actually trained in UBC. She did her PhD at UBC in Canada and also did her postdoctoral training in New York at Memorial Sloan Kettering Cancer Center. So I guess my last question would be, maybe can you share with our younger generation of audience as being trained overseas and also, but growing up in Hong Kong and being a faculty member in Hong Kong, what do you think is the differences, in terms of that?
In Canada, is training in Hong Kong. I study in quite conventional local schools. So with like maths coming very important, and not much room in classes or outside the curriculum that you can explore or do experiments or ask questions or find get some activities that or questions that you really like and you want to know more. So when I was doing my PhD, my mentor, which I show in the picture, Dr. Nellie Alsberg, she really inspired me a lot. So she's similar to that she trained as a clinical doctor, but turned out to be a basic scientist. And I still remember one thing she did, she always tried to learn new things. When she took me as a student, she already passed 70 years old. So, but she's still very active and being passionate to do research. I still remember in those days we used Mac, right? So for Canadians, a lot of time. And she decided to change her to a new computer. And she realized that if she buy an IBM at that time, which is a PC, that she can buy the same price, can afford 2 PC rather than just 1 Mac. So she decided that she would learn how to use PC at that time, so she can buy 2 computers, one for herself and one for the lab. So that's something like I think quite inspired me, like in the way how I as a person, or also in the way like how I work as well. Yeah.
I find it very interesting because I think it's oftentimes that it's the small things that you don't know that you're doing will make an impact to someone else's life.
Yeah.
So with that, thank you very much, Professor Wong,
Stephanie,
for your very insightful sharing. So I'll move on to our very last speaker, who is Professor Zhang Jianhua. Professor Zhang is currently an Associate Vice President of Global Research Collaboration and also a chair professor in the Department of Biology at Hong Kong Baptist University. So before that, Professor Zhang also served as a Dean of Science at Hong Kong Baptist University from 2017 to 2020, and was also the Director of State Key Laboratory of Agrobiotechnology at the Chinese University of Hong Kong from 2012 to 2017. So Professor Zhang is actually a very internationally renowned scholar and an expert in the area of plant stress physiology and water-saving cultivation of field crops. He is been recognized by a lot of awards and honors, including the State Natural Science Award twice, and also the State Scientific and Technology Progress Award from the State Council of China. He is also one of the highly cited researchers, and so it really attests to his scientific accomplishments. So without ado, may I invite Professor Zhang on stage now to share his talk, which is on China's food security and the sustainable use of water resources. Professor Zhang, please.
Okay. Many thanks to Stephanie for your nice introduction. Also, thanks to Hong Kong Laureate Forum to invite me to come here to share my idea with our young students, and it's a nice opportunity. I am a teacher. I always like to talk with students, so this is a good opportunity. We can have the platform even bigger than my classroom. So talk today I would like to give is, we say here, I put here, food security. Food security means, do we have enough food to eat? And here I emphasize China's food security as an example to show you that our food supply is not guaranteed. It's not guaranteed. They are limited by many resources and how you use the resources. And one key resource is water. So let me show you why I say so. So I focus on China's food production. And here show you the figure where population increase in China. This is the rapid population increase time from the 1950s to 1970s. And also, at the same time, the grain production in China, which is the main food, staple food we have. And the data is a bit older, but you look at the recent data, how much food we produce, how much population we all know in China, 1.4 billion. It's not, we do show this rapid changing time in China where the food security was always a problem. But whatever we have achieved, the achievement is population more than doubled from the 1950s to 1980s or later 2000. But food production more than tripled. So the conclusion is, or reality is, we have more food than our ancestors. We are well fed now than many years ago. So that's a big achievement. Then we may ask a question here, how we did that? Because if you read the history books, hunger, famine was always a problem in China's history. Many dynasties was overthrown just because hunger and people rising, and basically these sort of things happens again and again. So I tell you here, one thing here is how we grew our food. I say food production in agriculture is supported by three key resources. One is, of course, land. Land that can be used for growing food usually should be flat land. Two, the water. Agriculture use of the water is more than 70% the fresh water we have. So that's the major source of the water use in agriculture. Of course, the third resources for food production is temperature. So temperature, land, water are the three major sources for agriculture. If you have three, you can grow crops. Now here, what I try to show you here, the water, you need a certain much of the water to grow crops. And here, the green area means you have enough water, which is rainfall every year is more than 400 millimeters, which is enough to grow at least one crop. But below, water below this green area is yellow or red area. This means rainfall is less than 400 millimeters. That means not enough for growing crops. There may be enough for some grasses, some shrubs, but not for big forest, not for agriculture. So you will not not feel surprised to see where are the crops are grown in China. So here the greener means the more crops you grow, the yellow means very little crops you can grow. And in northwest of China, you have spots of this green area. That means we call oasis. Oasis means the water flows in the middle of the desert. Then people living there, they have enough water to grow crops. Then they have some trees. But the water is precious, of course, for this oasis agriculture. So China, we do rely on eastern part of near the coastline to grow most of the food. But do remember, in the southern China, you see the greener become lighter. This means we are very mountainous area. Then the land is not flat, so it's not good for agriculture. So we relying on the northern China plain, this Yangtze River basin for food production mainly. So this I actually in last here I just give you how limited we are. China, we have 5% water resources worldwide. We have 7% arable land, but we have to feed 19% the world population. That means China's food security is always threatened by the lack of land, lack of water resources. We have to use every precious drop of the water to grow our crops. Now here, this data showing you our food production increased over the years, mainly because we used more water for irrigation. So more irrigation area expanded, more food production we have. So that means their manufacturers, of course, contributed to our food production increase over the years. But one key factor is the use of the irrigation. So use water for growing crops is not wrong. It's always a survival for human beings over the years. But in many areas, you will find overuse the water for agriculture can be a problem. One of the problems in the area is in northwestern China. Northwestern China is have the inland river system. The inland river system means the water going to the desert from the oasis. You are not going to the ocean, so the water is used inside, cycled inside. Overusing the water in the oasis agriculture, you can have the problem. Now here showing you the Gansu province, one of the key northwestern China province. The food production over the years increased very much, and also the drought problem also become more frequent. The achievement for this food production in Gansu province, again, is because we use too much water to grow the crops. Now here, showing you the Gansu province. China's map here, Gansu province actually is a very long and narrow strip in China's map. And in the middle of the Gansu province is an area we call Hexi corridor. It's Siyanghe River. Now here is high Chining Mountain, very high mountain, glacier water comes down. And the water from a river going down end up in the Mingqing oasis in this area. So this greener area means crops are growing. So this is geography for the Mingqing area. The problem is because over the history, we were always on farming, so we try to grow more crops. So the cropping area has been increased very rapidly, especially in the upper and middle reach. So this caused the problem in the lower reach. The water for the mountain is changing a little bit, not that much. But you look at the lower reach, how much water you receive in the lower reach in the Mingqing oasis is declining. In the early 2000s, the river is almost dried up. There's no more water going to the lower reach. So this caused a huge problem in terms of ecology. The problem is, you look at this desert, newly formed. If you look at it in the inside desert, you see the shells there. The shells were living initially in the wetland conditions. But today, the wetland dried up, become sand dunes, and only fairly tough reeds plants still stay there. This reed plant is here, but reed plants are adapted to desert conditions and follow this moving sand. So this is a situation we developed because we were not properly use the water. And the end result is very familiar. In this area, people have to move out, villages abandoned, and the desert dust storm frequent several times in the year. So that was a horrific situation 20 years ago. The good things I should tell you, things has been reversed a bit, but we are getting better. There are many things we have done to achieve that. So that was a story showing you in area you have a certain amount of water. You have to use it properly. How much for crops, how much for ecology, how much for grasses, how much for trees. It should be well calculated. Otherwise, you have disaster. The second area is the major food production area is in the North China Plain. Northern China Plain is the our country's most wheat and cotton, maize are produced. They're all talking about 1/3, 2/3, to half in a number like that. And the problem is, again, we are overusing the water. This area, the water overusing, actually is because we are using underground water too much. And this showing you how much land irrigated area you increase, and actually how much over-exploitation of the underground water for the irrigation. And this, again, caused some unexpected consequences. Here, showing you, yes, over the years, the wheat and the maize production has increased. That's great. We said more than tripled. People were eating much better in this area. But if you look at underground, the water table has been dropping almost 1 meter every year. So this over-exploitation of the water in northern China Plain caused the river dry up. We used to have ship. We have ship to Beijing, but no more, not anymore. So this area, you're showing this picture, this red area showing how much water was depleted underground. And this underground water is precious, cannot be replenished easily. And there are lots of news reports. People were writing lots of articles telling you China's northern China Plain is drying up. The water table dropping down too quickly. And the farmers overusing the water for their crops. The reason I tell you, a fairly basic reason why we were overusing the water in northern China Plain. I said in the earlier picture, yes, this area is a major food production area. And the annual rainfall is about 600 millimeters, which is enough to grow 1 or 2 crops per year. The trouble is, over the years, we try to increase our production. And we grew more crops, more wheat, more corn. And this put the 2 crops together, they need water more than 600 millimeters. So more than annual rainfall. So the supplemental water has to come from underground. So that is the reason we have this problem. Of course, this situation has attracted attention. But this is just more data showing you, if we carry on like doing this, the northern China Plain will become inhabitable because we're talking about the modeling in 100 times, 2100 times, the temperature, they call this wet bulb temperature, will be too high. In the summer, you cannot carry on using this much water for irrigation. And this temperature will be too high for human beings to stay there. So this is an alarming signal to us. We cannot do that anymore. And we have to change our mind, how to think about what we call sustainability. It's a big issue. Sustainability is the key word I like to push for you. And there are, of course, lots of natural resources, and there are many natural resources. We initially use them. A good example for people misuse of natural resources is in the good story is the East Islands. You know, the East Island is sitting in the southern Pacific Ocean. The nearby land or islands is several thousand miles away. So it's a very, very unique geographic location. And because about 1,000 years ago, Polynesians arrived on this island. The island was full of forest. So people initially got there, maybe 1,000 people by chance got there 1,000 years ago. They didn't have an advanced ship. And they got there, they found lots of trees there, so they had a good life for many years. They're cutting down trees, dancing, they carved up the stones, make these stone statues beautiful, still there. The trouble, yes, the disaster is the last tree was axed about 300 years ago. They cut down all the trees for anything, whatever. And then they suddenly found they don't have trees, they don't have wood. Then they couldn't go out for fishing. They have to stand on the beach to catch fish, which was swimming to them. So the population will be fighting for survival. They're fighting with each other for a little bit of resources. So finally, when European sailors found them again, about in 1877, that was a record in history. There are only less than only 111. That's exactly the number recorded. Only 100 people live there. But in the peak time, they had, see here, 17,000 people at the peak time. What was named there? They got a good history of recording there. So that means if you don't use the resources wisely, and you have disasters, that's a good example for us. We have to remember this lesson. So how in agriculture to solve this problem? I said agriculture already uses more than 70% the freshwater resources to us. Now how we can, when we secure our food supply, another way we can have good life and sustainably use it, the only way I will say is water-saving agriculture. There are lots of potential to save the water for growing enough crops. So we have to realize the inevitable trend is you have to design the irrigation with less water. And you have to do research or show the farmers which each crop, with the specific water amount you have, and how you can save water. At the same time, make sure the plants produce the same amount of food for you. So over the years, we have been doing many water-saving agriculture approaches, methods, and there are many such ways. I said different crops can be different. You can do different things, but ultimate purpose is to use less water. But what we found is very interesting. We did many things on this area, and I don't have time for tell you all the results, but I just tell you finally when the results we found very interesting is alternate wetting and drying method. That means the water are delivered not all the time, but in partial time. And we found we can grow rice with much less water and still achieve the high yield. And just give you the results here. If you use we call AWD method to grow rice, and you can save about 9% the water, and you can increase the yield even about 9%. You save the water by about 29%, 1/3, and the water use efficiency for how much water for how much grains you produce can increase by 50%. So this is things we can achieve to make water more use more sustainable. So I think I have run out of my slides. So.
Thank you very much, Professor Zhang, for your sharing of your very impactful research.
Okay.
So we'll have a 5-minute Q&A.
Thank you, Stephanie.
Just as a reminder, if there's any questions from the audience on site, please just feel free to raise your hand, and someone will come to you with a microphone. Okay. So may I ask, so you advocate water-saving agriculture, right?
Yes.
As a method to secure food in China.
Yes.
So, I mean, our planet now, right? It's so troubled by such adverse weather, right? There is droughts, heat waves, flooding.
Climate change, yes.
So what is your take on that and, about just the very adverse climate change that we're experiencing now and your research, and what you're trying to advocate? Any thoughts and shares?
Thank you, Stephanie, for this good question. Climate change is a hot topic. Everybody blames climate change for everything, virtually. And for the water, climate change brings some water, maybe more, to northern West China. Recently, people not unproven, but there are some indicators. But in most other areas, you see drought more frequently, more severe drought in area which should not be so dry. So this is a really problem. So I believe the topic I'm talking about is universally interesting to all the agriculture area. We should not taking our water supply rainfall is taken for granted. We have to be prepared for unpredictable disasters to happen. Yes.
Thank you. Actually, I'm also very intrigued, right, in how you actually started in the field in the very first place. I mean, of course, there's a very big knowledge gap in what you're doing now, and we need to find a solution. But just going into science, what made you go into this area of research?
That's again good question. I was born in the Jiangsu province in mainland China, and I was doing farming for many years before I got a chance to go to university. So when I had a chance to go to university, the first choice was agriculture college in China. The reason is I had too much difficulties. I had too much hardship as a farmer. So I thought I could do something to improve the agriculture efficiency. So I went to the agriculture college. I learned crop production, crop breeding, the agriculture things. Then after that, my college, the Chinese government, sent me to UK for doing PhD for more research. I focused on plant stress, water stress research, but I always bear in my mind how the agriculture is doing in China. So when I come back to Hong Kong, I got a chance to collaborate with my colleagues in the mainland doing agriculture research. So I picked up this water-saving agriculture because I realized it is a big problem. So I had collaborations all over China virtually. So we had many experiments. We tested ideas how to save the water. And I've been very lucky for doing this over the years. I got many, as you said earlier, many awards and well recognized. So I feel happy. I'm doing things I feel passionate and feel fruitful.
Thank you for sharing that very personal story. I think, again, it echoes back to, of course, Professor Zhang started with something that also he holds dear to heart. And I think that is the drive for everything, right? And as tested, he's very renowned in his area, and I think that shows how dedicated he is to his field. Any questions? Yes, please. The microphone is coming to you.
Yes, yeah.
Okay, thank you. First, thank you for your sharing. And I would like to ask, can you provide more details on how to use less water, but to farm and perform the agricultural activities, but the results are the same as when you use more water? Thank you.
Yeah, you have learned. You got my idea. The water resources is limited. We only have a finite amount of water. You have to, of course, it depends on different areas. If we have abundant water, like in Hong Kong, every year here we have 1,600 millimeters of water every year. If you have the water, you use it, no problem. But in areas with limited amount of water, you have to calculate. You have to do shrewd calculations, how much you use for crops, how much you use for trees, how much you use for anything else. So this depends on the area, different locations, different amount of water resources. You do have to have good planning, and then you will decide if you have, say, if you have food shortage pressure, you have to grow this much amount of water, amount of food. Then you have to do calculations. Indeed, China has been stretched to the limit in a way to produce enough food for 1.24 billion people. And we are actually import most of our soybeans, which carries water. All the food means lots of water behind. So we buy the food means we buy water resources. So that is something we also do. You can do something else, say manufacturing, televisions, whatever, in exchange for food. That's acceptable. Thank you.
Thank you. I think I saw another hand going up. Please go ahead.
I would like to ask, because we live in cities and we may not directly go to farm and grow crops. So is there anything that we can do in our daily lives that can help the problem of water loss in China?
Well, that's a very good idea. Indeed, everybody can contribute to sustainable use of resources. In Hong Kong, we do have enough water, but our water is supply for drinking water is from mainland supply, which costs the energy and money. If we use less, of course, it's better. And also, we can contribute for resources like energy. We can use less energy, and that means also contribution for sustainable use of natural resources. There are many things we can do in our lifestyle, and also the idea we should pass to next generations, that everything has source, and all the resources are unlimited. We should not take everything as granted. We have to save everything. And you can enjoy life, as I always said to my daughters, but you cannot waste anything. That's a good idea we should have.
Thank you very much, Professor Zhang, for your sharing. Thank you.
Thank you.
Thank you. Thank you, Professor Zhang and Dr. Ma. Thank you very much. Please be seated. And thank you, Dr. Ma, for drawing the conclusion that we always choose to work in areas that are close to our hearts, just as what Professor Zhang did. He did some of his research in Gansu, his hometown, and that's why. Thank you once again. And ladies and gentlemen, time flies and this seminar has come to an end. And on behalf of HKLF, we'd like to take this opportunity to express our heartfelt gratitude to our distinguished speakers and moderator again for taking time off their busy schedule to make this seminar happen. Let's give them another big round of applause.